Hello everyone, welcome to Midha Physiology blog. I am Dr. Sonam and today we are on third video of the renal physiology. In last class, I had given you a brief introduction about a nephron. So here you can see that this all structure from the initial dilated portion to all tube-like structure, it is a nephron. Now let us see this initial dilated portion more detailedly today. Okay, so we had also came to know that this initial dilated portion is called the Bowman's capsule. Now in this Bowman's capsule, the blood supply is being coming from the afferent arteriole and it is dividing into capillaries, and via these capillaries only the blood would be filtered out and it would reach towards the Bowman's space, and then it would reach into the lumen of the tubular portion of the nephron so as to form the new urine. So today what we are going to see is how does the filtration of blood occurs through this capillary, because when we are talking about the capillary itself has a layer, and we also came to know from the last class that the capillary outside is also covered by a layer of the epithelial cells which we call the visceral layer. So when the blood has to pass from the capillary into the Bowman's space, it obviously has to pass through many layers. Okay, so how that filtration is occurring, that we would see in today's video. So the objectives in this video are to know about the glomerular membrane and to know about the process of filtration of blood. So the glomerular membrane, it has been defined as the structure which is intervening the passage of blood from the capillaries to the Bowman's space. What does this mean? So to understand this thing, let us look at this diagram again. Here, this one is the afferent arteriole. The blood is coming from the afferent arteriole and it is going into the capillaries. Now from the capillaries, the blood has to be filtered out to reach the Bowman's space. Now, as the blood has to travel from the capillary into the Bowman's space, it has to travel through the many layers. The layers formed from the capillary itself and the layers formed from the visceral layer of the epithelium. So these layers together is making the glomerular membrane. So you can see this concept more clearly in this diagram. Here you can see, so these are the capillaries, right? The arterioles, it is dividing into the capillaries. So these are the capillaries, and from these capillaries the blood has to be filtrated into the Bowman's space. Now, when you are talking about the capillaries, remember that the capillaries in our body, they have the single layer of the endothelium only. So firstly, the blood has to pass through the endothelium, which is making the wall of the capillary. And these endothelium, they are rested over the endothelial cells. They are lying over the basement membrane, which we call the glomerular basement membrane. So from the endothelium, the blood has to come out and then it has to travel through the glomerular basement membrane, and outside the capillary now, these capillaries are again being wrapped or surrounded by the epithelial cells, which is continuous with the parietal layer of the Bowman's capsule. Now, since this epithelial lining is covering the capillaries, we had called it the visceral layer. And remember, this visceral layer epithelium, they have become modified. Okay, they have become specialized because these cells, here you can see this typical epithelial cell, it has developed the food process. Since this cells has now started to develop the food process, you can call this cell as podocyte also. And in between the food process, here you can see a slit, okay, a tunnel like structure from which the filtrated blood has to travel. So altogether, when the blood has to come from the capillary into the Bowman's space, it has to travel from the endothelium layer and then the Bowman's basement membrane and then through the food processes of the podocyte. So these are three layers. These three layers together make up the structure which is hindering the easy filtration or all filtration of the blood into the Bowman's space. And these three structures together is making up the glomerular membrane. So here, this picture is being enlarged again. You can see this is the capillary lumen in which there is RBC, these are the WBC, and then this capillary wall is made up of this endothelial cells. And here the another peculiar feature you can see is the endothelial cells, they are not continuous. There is a gap between the endothelial cells, and this gap is around 70 to 90 nanometer diameter. And since there is so large gap between these endothelial cells, you also call this endothelium as the fenestrated endothelium, and this kind of capillaries as the fenestrated capillaries. Through this gap, the plasma can easily be filtered out except the plasma protein. So water, salt and the sugars and other electrolytes can easily now filter outside, and then it has to travel through the glomerular basement membrane or the basement membrane in which the endothelial cells they are lying. Now this glomerular basement membrane, they are made up of various kind of protein, and this layer, this basement membrane itself can be divided into three layers. Okay, the one which is nearby to these endothelial cells or which is internal to the capillary, you can call this lamina interna, and then which is facing outside, you can call it lamina externa, and in between the thicker portion, it has been named lamina densa. And now the plasma, after being filtrated from this endothelium and after it is going from the glomerular basement membrane, now it has to travel between the food processes of the podocytes. And I again repeat here, podocytes, these are just the modified epithelium which is surrounding the capillaries. And these food processes, they are not continuous in structure. These food processes of podocytes, they even have gap in between them, due to which they are forming a tunnel or they are forming a slit through which the filtrated plasma would now travel. And this slit is called the filtration slit. So the structure in the glomerular membrane can be divided into the capillary endothelium, the glomerular basement membrane, and the specialized epithelium of the Bowman's capsule, which we call the podocytes. So these are the three layers from which the blood has to pass so as the filtration can occur. Now, I have taken this diagram from Ganong 24th edition. Here you can see how these all structures are aligning. Okay, so these are again the capillaries. This is the first layer through which the blood has to pass, that is the endothelial layer, and then the second one is the basal lamina, and outside these all food processes, these are the food processes of the podocyte. Okay, here you can see this green color, this is the podocyte, and it is having so many finger like projection which you call the food process, or due to this food process, this epithelial cell itself has been named podocyte. Okay, and the gap between this food process is around 20 nanometer. So you can see, so this food process and this food process, the gap is around 20 nanometer. Due to this gap, there forms a tunnel or a slit like a structure. So this slit is called the filtration slit. And again, what they have found is, in these food processes are again lined by some fuzzy material. Okay, the proteins like the polyanion, which is also called the siloprotein. Okay, these proteins are lining the food processes of the podocyte, due to which they are again repelling the negative charged molecule which is coming from the blood. So overall, here we can say that from the capillary lumen, if the blood has to be filtrated into the Bowman's space, the plasma protein, they cannot be filtered. The larger structures like RBC, WBC, platelets, they cannot be filtered. However, the water, sugar, the salt and other electrolytes can easily be filtrating into the Bowman's capsule, the Bowman's space. But even in that electrolyte also, the negative charges are again repelled back. Okay, so the traveling of the positive charges, the filtration of the positive charges is comparatively easier here. So these all things are again summarized over here in this slide. You can see the first layer of the glomerular membrane through which the blood has to be filtrated is the capillary endothelium, in which the endothelium they are fenestrated with a gap of 70 to 90 nanometer, which allows the plasma filtration but with the retention of the plasma proteins and the blood cell. Then after, the blood has to pass through the basal lamina where the capillary endothelium lies, and this basal lamina has been divided into lamina interna, which is near to the capillary endothelium, and then which is facing the layer which is facing outside towards the podocyte, you call it lamina externa, and the middle layer you call it lamina densa. Then after, the third layer through which the blood has to pass for the filtration to occur are the specialized epithelium of the Bowman's capsule. Why are we calling it a specialized epithelium? Because the epithelium, it has developed the food process, due to which we are calling it podocytes, right? So the visceral epithelium, which is covering the capillaries, they are modified with the development of the food processes, and this specialized modified cells, we are calling it podocytes. The podocytes with having the food process, which we also call pseudopodia, they are forming the filtration slit along the capillary wall, and this filtration slit are having the food process is having the gap of around 25 nanometers. So the filtration slit is around 25 nanometer, and the fuzzy material are also present on the surface of these food processes, which are the glomerular polyanion, which are repelling the negative charges. So these are the three layers of the glomerular membrane through which the blood has to travel for the filtration to occur through the capillaries. So here you can see it again, the afferent arteriole dividing into the capillaries, and then these capillaries are again now draining into the efferent arteriole, right? So here from the capillary, when the blood is traveling, or when the blood transverses all those three layers, the capillary endothelial layer, basement membrane, and then the food processes of the podocyte, then only the blood would be filtered out into the Bowman's space, right? Now here, the other thing also you can see is from the afferent arteriole, as it is dividing into the capillaries, remember that in a Bowman, in a nephron, there are around 20 to 40 loops of this capillary. Okay, so there are all together so many capillaries, around 20 to 40 in number, and these capillaries are together found in different 6 set of the capillary loop, and each of this loop is having this 3 to 6 number of the capillaries. So altogether there will be around 20 to 40 capillaries in this nephron, and these capillaries are also having connection with each other, or the capillaries, they are having anastomosis with each other. Okay, and then here you can see, so in the nephron there are around 20 to 40 loops of the capillaries with anastomosis. Okay, and this arrangement is very favorable for the maintenance of the high glomerular capillary pressure, that is 45 millimeter mercury. So here you can see that the blood which is coming from the afferent arteriole, as it is going into the capillary, the hydrostatic pressure of this blood is comparatively higher than the hydrostatic pressure of the blood in other capillaries of our body. So in other capillaries, the hydrostatic pressure would be around 25 millimeter of mercury, but in the glomerular capillaries, the hydrostatic pressure is 45 millimeter of mercury. So this increase in the hydrostatic pressure in the blood of the capillaries is very much helpful for the filtration of the blood through this capillary. Okay, and this filtration under this high pressure, we also call it ultrafiltration, and the product of the blood, or the filtered product of the blood which is formed after the ultrafiltration, you call it ultrafiltrate. Okay, so eventually from this ultrafiltration, the ultrafiltrate would be formed in the Bowman's space. Now the question over here is, why does the glomerular capillaries, they are having higher hydrostatic capillary pressure? They are given two different kind of explanation. Okay, in some of the books they have written that the afferent arteriole, the arteriole which is supplying, which is dividing into this capillary, the afferent arteriole, they are comparatively thicker and shorter. Now, since they are thicker and shorter, the blood which is coming from the afferent arteriole itself is carrying the higher hydrostatic pressure. Now, the another explanation over here is the blood which is draining from the capillaries is over the efferent arteriole. And remember that in other part of our body, the capillaries, they are basically draining into the venules, right? The venules, these are the low pressure system. That means the pressure in the venules, they are lower. But since over here, the blood has to drain over the arteriole only, that is the efferent arteriole only, the efferent arteriole is having higher resistance, due to which the blood to pass from the capillary into the efferent arteriole, it is not so easy, due to which the blood would have the higher hydrostatic pressure in the capillary itself. So in either way, maybe because of the thick or short afferent arteriole, or maybe due to the higher resistance offered by the efferent arteriole, the hydrostatic pressure which is seen in these capillaries, that is higher than the hydrostatic pressure in the capillaries of other part of our body. So this hydrostatic pressure in the nephron, in this capillaries, that is 45 millimeter of mercury, and the filtration which is occurring under this high pressure, you call it ultrafiltration, by which the ultrafiltrate are being produced, okay? And this ultrafiltrate from the Bowman's space, now they would go into the lumen of the tubular part of the nephron. So here again you can see that in the systemic capillaries, the pressure, the hydrostatic pressure is 25 millimeter of mercury, but in the glomerular capillary it is 45 millimeter of mercury. So this filtration under this high pressure, you call it ultrafiltration, with the help of which there is the formation of ultrafiltrate. So this ultrafiltrate, it carries all the constituent of the plasma, okay? Mostly this constituent should be neutral with the diameter of around 4 nanometer, but these constituent does not contain the plasma protein, the blood cells, and other component which are more than 8 nanometer diameter. So with this we came to know that how the blood is being filtered from the capillaries into the Bowman's space, and how the pressure, the higher glomerular capillary pressure or the hydrostatic pressure of the glomerular capillary is helping in the filtration of the blood. Now, apart from that, in between this capillary, we are even seeing some another kind of cells, okay? So these cells seen between the capillaries, these are the mesangial cells. Now, these mesangial cells are actually the parasite-like cells. Now, you may wonder, what is this parasite-like cell? Actually, in our body, in the very minute vessels like the capillaries and venules, where the smooth muscle layer is smooth muscle layer of the vessels, they are lesser or absent, what we can see is, this is the picture of a capillary, okay? So here there is the endothelial layer, and this endothelium is now being rested over the basement membrane. Now in this basement membrane there is another cell, that is the parasite, okay? So these parasites are basically found in the basement membrane of the capillaries, and they are in close proximity with the endothelium. Now what is the function of this parasite which is located in the basement membrane is, whenever there is any damage to the endothelium, if there is the repair of the endothelium has to occur, or the angiogenesis process has to occur, or if there is some cellular debris, those all will be even cleared by this parasite, okay? Now the another very peculiar feature of this parasite is they are contractile in nature, due to which if the parasites they are contracting, even the capillary itself would start to contract, and it is also now controlling the blood flow through the capillary. Now similar kind of the cells, that is the parasite-like cells, are also found in between the capillaries of the glomerulus, and this parasite-like cell we have called it mesangial cells. So here you can see this again. So this is the glomerular capillary which is formed from the afferent arteriole, right? So if you enlarge this picture, here you can see, right? So if this is the glomerular capillary, this red color portion, these are the endothelium, right? And this endothelium, they are lining in this purple color lining. This is the basement membrane, and outside this basement membrane, here you can see the food process of the podocyte. Now you see that the basement membrane of this capillary is even being sheared by the basement membrane of another capillary, right? So the basement membrane is like a sheath which is being sheared by the capillaries over here, because the capillaries, they had anastomosis. And in between this basement membrane, here you can see this another cells, group of cells, and this group of cells, they are the mesangial cells. So these mesangial cells, which is seen inside this glomerulus, you call it intraglomerular cell, mesangial cell, and sometimes these mesangial cells are even seen outside this glomerulus or outside this capillaries, in between the afferent and the efferent arteriole. And this kind of mesangial cell, you call it extraglomerular mesangial cells. Okay, so they can be seen outside and they can be seen inside also. In either way, what they are doing is, if they are present inside, by their contractile property, it is causing the contraction of the capillary. If there is the contraction of the capillary, the blood flow through the capillary would be decreased, and the glomerular filtration rate would also be decreased. And sometimes when the immune complex, they are coming, or the cellular debris, they are coming, they are even being taken up by these mesangial cells. And while talking about this extraglomerular cells, which is seen outside the capillary or in the vascular pole of the renal corpuscle. So here, when you're talking about this vascular pole, you remember that this is you can recall that this is the renal corpuscle, right? So renal corpuscle is basically the Bowman's capsule together with the glomerulus, or together with the capillaries, network of capillaries. Now this renal corpuscle, at one end, it is starting the tubules, the proximal convoluted tubule, loop of Fendley, and other part of the tubules. So this pole is the tubular pole, and the pole through which the vessels, they are invaginating the Bowman's capsule, this is the vascular pole. So here, these extraglomerular cells, they are present in the vascular pole of the renal corpuscle, but outside the glomerulus, and is present between the afferent and the efferent arteriole. Okay, so the role of this extraglomerular mesangial cell is not so clear, but they have found that they may be related with the autoregulation of blood flow in the kidney, or the literature has even found that it is also associated with the release of the erythropoietin. Okay, so these cells together, when we are talking about the glomerular filtration, the filtration of the blood from the capillary into the Bowman's space, now this filtration can even be regulated by this another group of cell, which is the mesangial cell. Okay, so mesangial cells are basically lying between the capillary, endothelium, and the basement membrane. So let us see this thing again. The mesangial cells, which are the pericyte-like cells, can be intraglomerular mesangial cells if these are present between the capillaries inside the glomerulus, or it can be the extraglomerular mesangial cells if it is present outside the glomerulus between the afferent and the efferent arterioles in the vascular pole. So overall, its function can be summarized as the regulation of the glomerular filtration, and this function is very important, remember, because when the mesangial cells, they are contracting, they are also causing the contraction of the capillaries, right? The other functions, they are taking up the immune complexes, in which these mesangial cells, which are the pericyte-like cells, they are acting as the phagocyte, right? And they are also associated with the secretion of the extracellular matrix. So here you can see this picture again to get clear. This is the capillary, these are the blood, right? So this is the blood without the nucleus. You know that this is RBC. So obviously the blood has to be filtrated from this capillary into the lumen of the Bowman's capsule, that is Bowman's space. So it has to travel firstly through this endothelial layer, which is fenestrated, and then it would go through the basement membrane, and then it would go through the food processes of the podocyte. And this whole thing can even be controlled by another group of cells lying between the basement membrane. These are the mesangial cell. So again you can see this thing. So this picture is again from the Ganong 24th edition. Here these are the capillary, right, through which the blood has to travel. So this, the inner portion, this thin red color portion, these are the endothelium, and then the grayish portion, this is the basement membrane, and the cilia like structure, these are the food process. Okay, the food process here you can see it is from the podocyte. Now if you look at this capillary which is having the anastomosis, you see that if this is the grayish color layer is the basement membrane, this basement membrane is being sheared by these two capillary, and in between there is the mesangial cell, right? And this mesangial cell between the capillary is the intraglomerular mesangial cell, which is helping for the regulation of the glomerular filtration by either contracting or not contracting the capillary. So let us revise what have we learned in today's video. The first thing which we discussed is about the glomerular membrane, in which we found that the blood has to travel through 3 different layers, and then only the blood will be filtered into the Bowman's space, and this filtration process is even being affected or regulated by another group of cells which is seen between the capillaries, which are the mesangial cells, right? So this much for today, guys, and I hope this video was also helpful to you. If you like this video, please like, share and subscribe to my channel, and if there is any queries, any suggestion, please do comment. Thank you.